Hi everyone, and welcome to our video about carbohydrates. What we're going to be doing in this video, which is actually the first of a series of videos on carbohydrates, is having a look at the monosaccharides. So what we're going to be doing is having a look at specification reference 2.1.2d, the ring structure and properties of glucose as an example of a hexose monosaccharide, and the structure of ribose as an example of a pentose monosaccharide. And do note you need to know the structural difference between alpha and beta glucose and the difference between a hexose and a pentose monosaccharide. First thing I want to do then, before we delve too deeply into the realms of structures of carbohydrates, is just to remind ourselves of what carbohydrates are actually used for. And we've got three main uses of our carbohydrates. So the first one is as a source of energy, and this would be something like glucose, for example, being used in respiration. And you're obviously going to do a lot more about respiration in Module 5. Now, glucose itself will go through that process of respiration, and as an end result, we're going to get some ATP, and ATP is then going to be utilised in a range of metabolic pathways. Second use of our carbohydrates is as an energy store. Now, depending on if we're talking about plants or animals, this will determine what that storage molecule actually is. So in plants, we'll be looking at starch. In animals, we'll be looking at glycogen. And finally, our third use of our carbohydrates is as a structural unit. And I've given you two examples here of cellulose and chitin, both components of cell walls in different organisms. So what we're really seeing here is how these carbohydrates can be utilised to actually build those larger structures within the different organisms. And cellulose is obviously our plant cell, chitin is in our fungal cell walls. So in this video, we're going to be looking at the monosaccharides. Now, if we actually break that word down, mono just means one and saccharide means sugar. So when we're talking about these monosaccharides, we are talking about single sugar molecules. So these are only made of a single unit, hence monosaccharide, one sugar. Now, because these are only made of that single unit, then they are the simplest form of our carbohydrates. And what we actually find is we can generally spot them from their name because their names are going to end in OSE. So glucose, ribose, etc. They all have that familiar ending OSE, which is our little flag saying this is a carbohydrate. Now, as we mentioned earlier, these monosaccharides are actually really quite important in terms of being an energy source in living organisms. And when we're looking at the names of these different monosaccharides, then we can, in some instances, identify the number of carbon atoms from the name. Because what we actually find is that we will see things like hexose, or we might see pentose, even in some cases triose. So these are three different monosaccharides that we've got, and again we're going to use our naming rules. First of all, we can see from the endings that they are all obviously our sugar molecules because they all end in OSE, and the first part here tells us how many carbon atoms there are. So when we've got hex, hopefully we can think of other words in our everyday existence that we've heard of that have hex in front of them. That just means 6. Pent, again, from everyday life, hopefully we know that pent means 5. And tri is 3. You can go to your maths knowledge here. Triangle, pentagon, hexagon. The same numbers, the same prefix there. So what we actually have is triose is a 3-carbon sugar. Pentose is a 5-carbon sugar, hexose a 6-carbon sugar. So we can ascertain quite a lot just from understanding those prefixes and suffixes in biology. So really do get to grips with breaking down these weird and wonderful words we like to use so that you get a better understanding, so that when you're presented with unfamiliar things, you can just unpick it and go, well, it ends in OSE, some kind of a sugar then. If we now think specifically about our monosaccharides and their properties then, One of the first things we need to remember about them is that they've got a large number of these carbon-hydrogen bonds. And this is going to be really important in terms of their ability to provide energy. Secondly, they are soluble in water and insoluble in non-polar solvents. So soluble in water, but not soluble in non-polar solvents. And hopefully from our earlier work on water, we remember that water is a polar molecule. So it's going to be a polar molecule. We'll have a quick recap on water because we've got our oxygen and two hydrogens. The hydrogens have that slight positive, the delta positive charge, and our oxygen has that slight negative, the delta negative charge. So because we've got a slight positive and a slight negative charge on the two different ends of the molecule, it's polar. So monosaccharides, soluble in water, insoluble in non-polar solvents. An important thing to remember is this ratio of the carbons to hydrogens to oxygens, and it is as simple as 1 carbon to 2 hydrogens to 1 oxygen. Now, if we know this ratio, then whatever number of carbon atoms they give you for a carbohydrate, we can work out its formula. So we've got glucose as an example. If we know that there is a monosaccharide with 6 carbons, then we apply our ratio. So we've got 6 carbons, hydrogens, there's 2 obviously to 1, so that means that we've got to do 2 times 6, which is 12, and then our oxygens at the end is the same as our carbons, so 6. If we were doing a 5 carbon sugar, we would have C5, H would be 2 times 5, which is 10, and O would be just 5 again. So by knowing that little ratio, we can work out the formula of any monosaccharide you could be given the number of carbons for. Now, when it comes to the actual structures of how these carbons, hydrogens and oxygens are all arranged, we need to remember that it's not just one way. Despite the fact that we may have glucose, which is C6H12O6, one of our old favourites from GCSE, it's not arranged exactly the same way in every single glucose molecule. Why would it be? Biology wouldn't be as fun and complex then. Instead, we find it can be arranged either in straight chains or in ring forms. So what we're really talking about here is something called an isomer. And isomers, quite simply, are molecules that have got the same formula but different arrangements of those atoms. So what we would have is we've got our C6H12O6, so we've got the same number of carbons, hydrogens and oxygens, but the isomers would be arranged in different ways. And what we're going to do is have a look at some of these isomers to give you an understanding of how this works. If we start off with the straight chain isomers, so we're sticking with glucose throughout this bit because it's an old favourite, so let's stick with what we know. Then what we actually find is we not only have a straight chain version of glucose, we have 2 straight chain versions of glucose. We have L and D glucose. So if we actually have a little look at them, then what we can do is if we start at the top, we can see that that bit at the top is exactly the same. If we look at the bottom, again, they are exactly the same. If we look at the middle section, we've got that little backbone of all the carbons running down, exactly the same. So where do we get the difference? Well, quite simply, if you have a look at where those different hydrogen and hydroxyl OH groups are arranged, then what we can see is we get a different orientation of them. So you can see on L-glucose that at the top there where I've just put the purple box, we've got a hydrogen on the right, whereas in the D-glucose, we've got OH, the hydroxyl group. So what we're really doing is we're flipping them. So you can see that in each of those cases, the hydrogen is on the opposite side in L-glucose to D-glucose, and therefore the hydroxyl is 2. Now, the biggies. These are the ones you just need to know, folks. So what we're going to find is, as part of your exam, you could be asked to actually draw the structure of alpha or beta glucose or describe the differences between their structures. So This is where I suggest that you actually just practice this, because there's no shortcut to it. You just need to sit there, draw them out until you can do it pretty much in your sleep, I'm afraid. So the ring forms of glucose have, again, two versions. We've got alpha glucose, which is the one on the left, and beta glucose, which is the one on the right. When it comes to just explaining the difference between these structures, then what we need to say is that the hydroxyl group on carbon 1, and be specific here, so when you're writing about it, just make sure you are saying carbon 1. So the hydroxyl group on carbon 1 is, and then depending on, obviously if you're talking alpha or beta, you'll be talking about above or below the ring. So the difference between them is the position of that hydroxyl group on carbon 1. So let's have a little look at these then. First of all, when we're looking at the structures, you'll notice that there is this oxygen in the top right, basically. Okay, so that's our starting point, if you like. So when we're actually going around, all of these little points where we kind of get to a join, they are carbons. So what we've got is carbon 1, carbon 2, carbon 3, 4, there's 5, and up top is 6. And it's exactly the same on the right, carbon 1, 2, 3, 4, 5, and carbon 6. So we've got our 6 carbons because glucose is a 6 carbon sugar. So whenever you're drawing this, always start by getting that initial ring structure, making sure you remember the oxygen in that top right position. Then what we need to do is obviously finish off our carbon 6 group. So you just need to add the H2OH onto that 6th carbon atom at the top. And then what we've got to do is get the hydroxyl group on carbon 1 in the right place. So in alpha glucose, it always goes below the ring, whereas in beta glucose, it goes above the ring. Then what we're going to do is put a hydrogen on the other end of that particular bond. For carbon 2, the hydroxyl group always goes below, and then you just alternate. As you can see, carbon 3, it's above, carbon 4 is below, and then you just put hydrogens on your spare bond, so to speak. So my suggestion is start with alpha glucose. Okay, so what you need to do is just get yourself a blank piece of paper, and then what we do when we're drawing this is you start with the oxygens. Now from there we can add in the carbons. You can draw them as carbons, or you could just draw them as the points as we saw previously. So I'm going to do the carbon atoms just so that you can see the other version here. So we just come round and we're putting in our carbons to make the ring first of all. So we've got carbon 1, 2, 3, 4, 5, and then we put in our carbon 6, and you might as well finish off that group as you go. So CH2OH. Now, because we're doing alpha glucose, then what we know is that the hydroxyl group goes below the chain here. So put that one in straight away, and then let's just put our other bonds in place so that they're nice and easily there for us. What we then do is we're going to put the other hydroxyls in. So carbon 2 is always below, then we go above, then we go below, and that's our hydroxyls in. Do notice how on this one here I've put the oxygen closest to the bond. If you put hydrogen and then the oxygen on the outside, your bonding is going to look wrong, because at the end of the day what you're saying there is hydrogen can make 2 bonds, which it can't. It can only make a single bond. So always the oxygen pointing towards your bond there. Once we've done that, any of the spare bonds, just stick a hydrogen on it. There we go, and don't forget the one in the middle there. Job done. If we were doing beta glucose, we'd do exactly the same thing, but we would have these two flipped. So practice. That is the best thing you can do to be able to draw your alpha and beta glucose perfectly every time. So here's our lovely alpha glucose, and we know it's alpha glucose because our hydroxyl on carbon 1 is below the ring. And this is going to be one of those key energy sources, and it's also a key component of both starch and glycogen. We're going to get very familiar with alpha glucose as we go further through the course. Beta glucose, we know it's beta glucose because the hydroxyl group on carbon 1 is above the chain. Again, energy source, but this time it is a component of cellulose. So you can start to see here how knowing your alphas and your betas is going to be important. Otherwise, when we come on to the other types of our carbohydrates, you're going to mix them up and lose the marks. One word of warning about when we are writing alpha and beta glucose before we leave this section. If you are writing alpha, you can either write the whole word alpha, or we can use the Greek symbol alpha. You cannot use an A or an A. Either of those is not getting you the marks. Just like for beta glucose, you can write out beta or the Greek symbol. You cannot have either a capital B or a lowercase b. No marks for those. And beta glucose is probably the one to be the most careful with, because when we look at our actual Greek symbol for beta, it kind of looks like a capital B just with a long tail on it. Now, my advice is exaggerate that tail if you're writing on an exam, because some of your writing isn't great, and if you're just doing this teeny little tail, is it a beta or is it a capital B? So really do the exaggerated tails just to make sure there can be no doubt in your examiner's mind as to what it is you are writing. If you think back to our specification reference we looked at the start of the video, we said ribose is another structure you need to know. Now, ribose is a 5-carbon monosaccharide, therefore using our ratio, we know that it is C5H10O5, 1 to 2 to 1, and the ribose itself is going to be familiar to us, hopefully, from RNA, ribonucleic acid. It's a component of that. And as we move further through our A-level biology, we will see it also cropping up when we look at ATP and NAD. So ribose is a component of RNA, ATP and NAD. You do need to be able to draw ribose as well. So the same kind of process applies to how we looked at learning the glucose molecules. Now, when we have a look at ribose, first of all note that the very top position is our oxygen. So from the oxygen, we then need to create our ring structure, which this time has 1, 2, 3, 4 carbons in it. And ribose obviously has 5, so our 5th carbon is the one that comes off the top as part of that CH2OH group. We then just need to place our hydrogens and hydroxyls in. Notice how that we've got a hydroxyl group above the ring on that 1st carbon, and then you've got below on the 2nd, below on the 3rd, and then you can just insert the hydrogens in those last spare bond positions. Practice drawing these out. That is the best advice I can give you. There is no shortcut, there is no kind of cheating way around this. It's just practice it over and over again until you know it. We also have another 5-carbon sugar, deoxyribose. Now, deoxyribose is the DNA sugar. So ribose in RNA, deoxyribose in DNA. And if we just have a look at the name deoxyribose, well, if we break it into the 2 parts, we've got ribose, which tells us that that is a 5-carbon sugar molecule because we've got O's at the end, so it's a sugar, and it's a 5-carbon because we're familiar with our ribose, hopefully. Deoxy just means basically less oxygen, one less oxygen. If we D something, we remove it, so we've removed an oxygen. So when we're looking at our ratio, it would obviously be 1 to 2 to 1, so we'd have C5H10, but because it's deoxy, we've removed one oxygen, it's only O4. So just go careful there. Now, when we actually have a look at it, we can see that the structure is identical to the ribose, with the exception of the hydroxyl that we had on our 2nd carbon. It's now just a hydrogen. So that's where our oxygen has gone from. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when more videos are uploaded. Head on over to the website where you can find a range of other resources to help you in your A-level biology studies.